So we would like to start, please. So the first scientific talk will be given by Professor Wolfgang Helfrich, and you can see the title, How to Get from Physics to Biophysics. Please.
Yeah, in the first place, I would like to thank Tel Aviv University for this prize. Now, this is the more formal thank you. The Raymond and Beverly Sackler International Prize in Biophysics. And my special thanks go to Misha Kozlov, who is not here, and to David Andelman. Without them, I would not be here. Now, apart from being a great honor, the prize is a reason for happiness and pleasure, because almost 40 years of my research were devoted mainly to fluid bilayers. And you all know that they are the matrix of biological membranes and sometimes called biological model membranes. I'm also very pleased that my former, not my, but the former Alexander von Humboldt fellows who joined me for quite a number of years in Berlin, one after the other, are here, and I'm talking about Ouyang Zhongkan, and I would like to mention Misha Kozlov as well, but I can mention him. Unfortunately, he's not here. Now comes the difficult part, which is not really scientific. I should tell you the task I was given by the prize committee, and you see it here. I'm supposed to give you a historical overview on the over 40-year evolution of soft matter physics of lipid bilayers and biological membranes. To be quite frank, I'm not able to give you a balanced account. Instead, I will talk about a mixture of two things. My rather turbulent entrance into biophysics and some highlights in the 40-year evolution from my perspective. I'm certainly unable to talk about what is hot today in the field of biophysics, even if it started perhaps from bilayer membranes. What I would like to recommend here is a history of liquid crystals with a title given here by David Dunmer and Tim Slockin. It is quite new and very readable. It's like a detective story, full of suspense and full of details about the scientists, personal details about the scientists involved. And this book includes the discovery of the bilayer structure of biological membranes and smectic soaps. And this is not so long ago. In fact, in 1925, two Dutch scientists studied red blood cells, separated the lipids from the rest, and looked for the area of the monolayer these lipids made on a water surface. And they could compare with the area of the red blood cells. And from this they got evidence that, red blood cell membranes, as far as their lipids, consist of lipid bilayers. Some people criticize this work. There are some errors in the estimates, but the errors cancel. And I think it is quite exciting. Another paper was published in 1939 by Stauff. This is the title. And in this study he looked at, with X-ray reflection, at the different micellar solutions. And when he had a lyotropic smectic liquid crystal phase, soap and water, he could tell from his signal that the lipids are, not the lipids, the soaps are arranged in a bilayer. Now I have no books to help me, and this was typical of my work. There were no books to help me. But what do I want to tell you next? About my development towards, first, how I moved as a student towards organic materials. I did my diploma thesis in Göttingen at an institute for nuclear physics, and I was not asked to do any experiments with radioactivity, etc. I was asked to build an NMR apparatus which was intended for exercises of the students. I had to travel to Munich to get a plan for the electronics, and okay, finally it worked. I saw beautiful wiggles of the proton resonance in water, and from there I'm too far. Sorry, something went wrong here. How to go back?
Is this back or forward? Okay. You want one more?
Yeah.
One more?
I don't know. Thank you. Where should I? This is forward, and this is. Yeah. This is forward. What is backward? This is backward. Simply here. Yeah. Yeah, this is the right slide, and from Göttingen I went to the Technical University of Munich to get my Dr. Reanat, as it is called in Germany, and again this was basically a nuclear institute with a lot of solid state physics too. It was headed by Leibniz, who had established just a few years ago the first nuclear reactor in Germany. I was given as a task to measure the conductivity of anthracene crystals, and I failed. After 2 years, I had found out that it is so low, at least 2 orders of magnitude lower than what my thesis advisor expected, that I couldn't measure it. And then, yeah, Peter Mark came along from Boston. He's was the son of the well-known professor of polymer chemistry at the Brooklyn Institute of Engineering. And he suggested to inject carriers into organic crystals. And so I did, and I measured space charge limited currents. Then I went to, after 2 years in Munich with some students and busy with writing papers, 3 papers on my thesis with Peter Mark, and still in German. Then I went to Ottawa, to the pure chemistry division of the National Research Council. There they had beautiful anthracene crystals. They had a very good technician who could grow centimeter-sized anthracene crystals. And my boss there, Bill Schneider, advised me to try double injection. He knew, as a chemist, he knew how to inject electrons. So I followed his advice, and everything worked beautifully, and we got not only current, but again, all right. We got not only current, but also a pale blue recombination radiation. Some people are telling me this was the first organic light-emitting diode. No, I didn't know the word at that time. We were thinking of illuminating wallpaper. I'm sure we were also thinking of television, but this was far, far away. From Ottawa, I went to Princeton, New Jersey, to join RCA laboratories as a member of the technical staff. And at first, I had a very, I would say, boring task, the mathematical solution of a heat conduction problem. And after I had written a report, I asked for more interesting work, and I was assigned to George Heilmeyer's liquid crystal display group, which at that time was hardly known within RCA labs. It was a still secret endeavor, but soon to be publicized. And I got a rather attractive charter to help them understand the physics of their displays. Them is electrical engineers and chemists and technicians. The trouble perhaps was I didn't know anything about liquid crystals at that time. I didn't even know the word. But shortly afterwards, I went to Munich for about 10 months to get my Habilitation. You know what it is. Today it's no longer necessary, but it was needed to be eligible for a professorship, and I made it as an option. I was offered it, and I made it as an option. I didn't intend to become a professor. I was afraid of lecturing, of teaching. When I returned to Princeton, I began to study available liquid crystals, articles, published some theoretical papers, 2 of them even in physical review letters. I felt Americans are very kind with foreigners. But eventually, I suggested to George Hallmeyer to try, in addition to the devices he had developed in his group, to try what today is called twisted pneumatic. And this was 2 times, if I remember correctly, in 1969 and in 1997. However, Hallmeyer rejected the idea. He didn't like that you need 2 polarizers and that the light has to pass these polarizers at least once, usually, for instance, television, twice, unless you have backlight. Then it's, no, 4 times, unless you have backlight. With backlight, it is 2 times. So the group was disbanding already. Hallmeyer was on his way to a White House fellowship, and there was nobody really interested. So I took the idea with me when I returned to Germany, or rather Switzerland. I had a very interesting offer from Hoffmann La Roche in Basel. They hired me as a liquid crystal physicist for reasons which I don't know too well. But they had a liquid crystal group, and I became a member of this group. And at that time, I had a vague idea that eventually I might end up in biophysics. And I didn't like the idea. So employment in Switzerland began in October of 1970. And I suggested immediately to try twisted pneumatic. And Martin Schad, an experimental physicist who was already there, built such a cell. It worked beautifully. And before the year, we applied for a patent and submitted a note to Applied Physics Letters. A few months later, there were first signs that my job in Basel was not secure. And in January 1973, I was given notice by the director. This was no surprise because Roche had diversified into many directions. They had too much money earned with Librium and Valium. And finally, they realized that diversification was not such a good idea, and cut it back again. I was one of many victims. All right, there was a consolation. The same day I got a phone call from a future colleague at the physics department of the Freie University of Berlin. I had been chosen for an associate professorship. Now, I developed the theory of bending elasticity of fluid bilayers while I was still at Roche. I had set up in the meantime, the good years, a theory of bilayer bending. The bending energy is a reduced and modified version of that valid for pneumatic liquid crystals. Sir Charles Frank found the final form in 1958. So I knew a lot now about liquid crystals, and I was also fairly familiar with Euler-Lagrange, with the Euler-Lagrange formalism. And this again is a consequence of work with liquid crystals, of the distortion of the deformation pattern in the usual cell between two flat plates. And I used the formalism to derive a shape equation, at least for cases of rotational symmetry. Now, at that time, Basel was a particularly good place for a start into biophysics. Joachim Selig at the new Biozentrum was doing his famous studies of the order parameter of the hydrocarbon chains in PC bilayers. And yeah, he offered me a 5-year position at the Biozentrum, but with three small kids, I preferred the position at FUB. It was rather sad because we had a house built according to our ideas with some American conveniences in a beautiful landscape, and we had to leave all that. There was a new opportunity at the physics department in Berlin. My colleagues permitted me to do experimental or to have a mixed group of experimental and theoretical students. At that time, this was unheard of in Germany, but Berlin was special, very liberal. Many of our people came from the US, and we also had plenty of money because we were an outpost in the East. But before I assembled a group of students who wanted to do biophysics for their diploma, I met Heinz Steuling, and Heinz Steuling was an assistant professor of theoretical physics. He later became a professor of physics at Kassel University, and he was able to write my second-order differential equation on a bundle of punch cards and solve it on an early computer. In 1974, we could precisely reproduce the red cell shape and later on calculate a catalog of vesicle shapes, some of them also known from red blood cells. In this process, we learned of competitors, so it was actually a rather hard thing to do. We learned of competitors who had failed. There was Canham, a theoretical biologist in the US, who had tried to simulate the red cell shape. by methods which were too primitive, and Nering and Saupe tried two. This is not published, and both groups left out spontaneous curvature and had no shape equation. Spontaneous curvature is very important here. It is an adjustable parameter. It allows you to vary your theoretical shapes. Others were close on our heels. Evan Evans, many of you will know him. He published a paper on the bending moment in red cell membranes in 1974. And perhaps the most dangerous were François Brochard and Lennon in France. They published in 1975 a paper on the flicker phenomenon of red blood cells. And they already used my theory, fair enough. But this work was based on a thesis, and I think they must have had developed some ideas during the thesis. But okay. Now, now I want to mention some major events or major pushes for soft matter physics around 1970. The most important person here is Pierre-Gilles de Gennes, future Nobel Prize winner, not only a very good theoretical physicist, but also a unique personality with many interests, and a very helpful kind, unless he was absorbed by one of his problems, that you could meet him and he wouldn't see you. Now, this happened very rarely to me, but he was known for it in Orsay at the lab. And he once left me on the station at Orsay. We were waiting for the train to Paris, to the Collège de France, and we were with our wives, and when the train came, he moved along very quickly to first class. I took second class, but all right, he had to do some important work. Now, again, I'm on the wrong slide. De Gennes founded the liquid crystal group at Orsay only in 1967. Already in 1971 he began to move on to polymers, but he was still very active in liquid crystals as well. And he wrote two books during these years: The Physics of Liquid Crystals and Scaling Concepts in Polymer Physics. He had published a book on superconductivity before he did these things, and he published a fourth book with a co-author on wetting after these things. I would also like to mention two notable foundations in 1971, at least in the small German-speaking part of the world. They were important. One is the Biocentrum or Biocenter at Basel University with 4 professors of biophysical chemistry, and the other is the Max Planck Institute for Biophysical Chemistry at Göttingen with Manfred Eigen. Manfred Eigen studied both chemistry and physics. But there's always biophysical chemistry instead of biophysics. And part of the reason is that biophysics was the wrong word at that time. It was limited to X-rays in medicine. And this was not what these people were after. Also a few words about the USA and other countries where I'm not an expert at all. It seems to me that soft matter physics progressed there on a broader front, more groups involved, more directions, and often was triggered by applications. There are a few examples here. One is James Ferguson. James Ferguson was our competitor. I mentioned a competitor in connection with twisted pneumatic. That was him, and I was very much aware of the threat arising from him. He had worked on and invented cholesteric liquid color sensors at Westinghouse, I think. And then he went on to Kent State University. But of course, he would be interested in electro-optical displays as well. So I was afraid of him. I also mentioned Evan Evans. He's, or he was and still is, a professor of biomedical engineering, something we don't have in Europe or Switzerland. And yeah, which is an engineering approach. He published a book with Skalek on the elasticity of the red blood cell membrane. This was a little later. Another person who impressed me was Ora Kerem from Israel. She visited us, I don't know for which reason, in Berlin. And I knew she was a desalination expert, but then she turned out to be very interested in lipid bilayers and biological membranes. So it was a very nice encounter. And there were Perutz and Kendrew in England who obtained the structure of hemoglobin by X-ray diffraction. And some people were quite impressed by it, and my thesis advisor, Professor Nikolaus Riel, had a task for another student, which was to measure ultrasound absorption by hemoglobin. And this was in 1960 when he was given the task. So our supervisor, Professor Riel, was aware of what was going on. Yeah, then came a time of widespread interest in lipid bilayer and biomembrane soft matter physics. And now I am in a position I don't like at all. There's a list of names, and it may suggest some ranking. It doesn't, not at all. And it is also very unfair because all the persons listed here are listed only once, but often they did a number of these things, were involved in a number of these things. Yeah. Now I could talk a lot, but I think I should not spend too much time on it. But very soon there were quite a number of people whom I met quite often, and we had interesting discussions. And yeah, very many of them, of course, with Erich Sackmann, and then with Adrian Parsegian, with Reinhard Lipowski, too. But I think I had discussions with almost all of them. Okay, forget it. I mean, you know the names, or just keep, or if you don't, keep the names. Today, the situation seems to be different. To me, there seems to be a new direction of biomembrane physics of the classical kind, mostly statics, not dynamics. But even that is not true. This restriction is not true. I give here a few names and a few back again. Yeah. And a few abbreviated titles of publications of their work. And there are two people you could put together in France, two physicists, an experimental physicist and a theoretical physicist, Patricia Bassereau and Jacques Pro. And there's another group centered around Mikhail Kozlov, and he's a theoretical physicist, as you know. Here are the titles, and yeah, it doesn't really converge, but it goes into a particular direction, if you like, and that is shaping of membranes by proteins. This I find very fascinating. I'm old-fashioned, and I don't like very complicated calculations. I like tricky models. Okay. Here you see the full titles of the papers, and you see a long series of names. I mentioned Jacques Pro, theoretical physicist, and Patricia Bassereau, experimental physicist, and other people, and some of them were extremely important. For instance, Bruno Gu, who is a biologist, at least he's a bioscientist, not a physicist. And Okay, this is a mostly theoretical paper, the first one. And yeah, I find it very, very interesting and very new to me. There's another paper of the French group. You see Patricia Bazarot, again you see Jacques Pro, you see another theorist, Jean-François Joanny, who, if I remember correctly, was a graduate student of de Gens. And there's Bruno Gu again. But what is new here in my mind, you have no longer lipids only, and no longer lipids and perhaps a single protein. You have the interaction of, you consider the interaction of lipids with proteins, some of them even motor proteins. And yeah, what I like best here is, for instance, this paper, a review paper by Joshua Zimmerberg and Michael Kozlov, how proteins produce cellular membrane curvature, which means the curvature of organelles of any kind. The endoplasmic reticulum, for instance, and perhaps sometime mitochondria. I know I'm not fair now. I see best what I like most. But I would like to mention some other new directions. There's a theory of complexes of DNA. There's a mistake, DNA, of course, with lipid bilayers. This started quite a while ago, and more recently there is work by Jacques Pro and Frank Jülicher, the theory of dynamic processes in cells and tissues. We heard about it today. Entirely new structures appear. There are skeletons, and it's not everything is lipid membranes. And when you philosophize, and I philosophize a little bit about the new situation, then it seems to me that physicists are needed in the biosciences mainly as providers of technology, very sophisticated engineers perhaps, and as theorists. You need them both, but in the middle are the biologists or medical scientists, and they are tearing physics apart. I mean, I could still do everything in my group. I was in this lucky position, theory and experiment. Today, this would be utterly impossible. And that's the way things develop. And if you like, you can speak of a kind of globalization. Not the globalization of science as far as traveling is concerned, but the globalization in the sense that you can't separate the different disciplines as well anymore if you want to do interesting things. Yeah, thank you very much. And yeah, can you help me please if I don't understand them?
So if there is a question or two, but please speak up because both Professor Helfrich and me, we have hearing problems. Any questions? The young people, how to become a lawyer?
Yeah, I'm a piece of old furniture. I'm aware of it, and I told Misha that I've troubles with the subject I was supposed to talk about, but you were relentless, and he told me it can be a little less than 45 minutes.
Yes, Adrian, please.
In all these bending elasticities, people talk about spontaneous curvatures. And you hear people inventing spontaneous curvatures for bilayers. Sometimes it's well-founded, sometimes it's unconscious that they added to the energy. What's your view of how you have good criteria for choosing spontaneous curvatures?
So the question was about spontaneous curvature. Yeah. So sometimes it uses a parameter, sometimes it has a deep notion. What is your view?
Yeah. In connection with red blood cells, our basic approach is the fact that the two monolayers can slip past each other. And all right, a deformation produces spontaneous curvature because of that. But the spontaneous curvature is not a local phenomenon in this model. There's just one spontaneous curvature for the whole cell. And we adjusted the spontaneous curvature until the figures were right. And we were lucky because Evan Evans studied red blood cells as an experimentalist too, and he found a very strong shear elasticity. And you could expect that shear elasticity destroys the usefulness of our calculations. But unfortunately, it is a viscoelasticity. So after a while, shear elasticity vanishes, and you are back at our simple model. So you need good luck from time to time.
Any other questions? If not, let's thank Professor Helfrich again.